All right, so chapter 5, part 2 is looking at membrane function. So the title of this chapter was The Working Cell. In the first lecture, we talked about energy, ATP, and enzymes. And then part 2 is the function of the cell membrane. So let's look at um, different kinds of movements of small molecules through the membrane. So let's review, before we really get into it, the membrane. So remember that the cell membrane is made of a phospholipid bilayer. The hydrophilic heads face the outside in gray. The hydrophobic tails are inside in the middle. The membrane is going to be selective. Remember the selectively permeable barrier as to what goes through this membrane. Not all molecules can go through the membrane. If you look at this picture, nothing's actually going through the membrane itself. They're using proteins to come in. Here's another channel protein. So there are some molecules that are small enough and they can dissolve in fats, because one of the things about the membrane is that it's a fatty interior, right? We have these fatty acids. This is non-polar. So molecules that move through the membrane have to be able to dissolve it through this non-polar area. So they have to be small molecules or they have to be non-polar in order to go right through the membrane. So we're going to look at different kinds of movements across the membrane. The first kind of movement is called passive transport. The word passive means that there's no energy required. When we talk about energy in the cell, we're talking about ATP, okay? The kinds of transport that fall into passive is diffusion, facilitated diffusion, and osmosis. Then there is active transport. Active transport means that you have to use energy. And again, energy means we're using ATP. And then the last kind of transport is endocytosis and exocytosis. Also energy is required, but it's a little bit different than the others. Okay, so we're going to start with diffusion. So what is diffusion? So let's look at our outline. Diffusion and facilitated diffusion. Both are considered passive transport. Passive movements means that molecules will move down their concentration gradient until they reach equilibrium using no energy. That's a little, maybe a little, but no energy is being used. No ATP is being used. So let's take a look at what's happening. This side of this picture has a lot of molecules. Dye, whatever it is, it doesn't matter what it is, but they're molecules, right? And there's a high concentration. There's a lot of molecules on this one side of the membrane. There are little holes that these molecules can move through. So these molecules are going to move randomly, right? Because all molecules and atoms, there's random motion. Everything's moving. So because of the random motion, they're going to move through to the opposite side. So what's interesting is that diffusion is the movement of molecules from a high concentration towards a lower concentration until they reach equilibrium, right? So the diffusion, the net diffusion means that we're going to have these molecules mostly move in this direction. Net diffusion is moving this way. Net diffusion is moving this way. Now, the molecules will move back and forth because they're randomly moving. They don't have a thought to say, like, I want to go over there, right? They're just bouncing and wiggling and vibrating. And some of them might pop over to the other side, but over time, the net diffusion still moves most of those molecules over to the other side until it reaches equilibrium. So when there's a balance of molecules on both sides of this this barrier, right? This is called equilibrium. There's the same amount of molecules on both sides. Now, the molecules will still move back and forth, back and forth. They're still moving randomly, but there's no net movement. There's no push for the for the molecules to mostly go to one side. Okay, so molecules will diffuse down their concentration gradient. When I say down, I mean from high to low, okay, until they reach equilibrium. And then there's a backy-forthy thing going on, but no net movement anymore. What's really interesting is that each molecule will diffuse down their own concentration gradient. So look, now we have two different molecules. We have purple and we have red. The purple molecules have a higher concentration on the left side, and they're going to move towards the right side, right? They're diffuse down their diffusion gradient to the right side. Whereas the red molecules have a different diffusion gradient. They have a higher concentration on the right side. They want to move over to the left. And so they're both going to diffuse according to their own gradient until they both reach equilibrium. And this is passive, so there's nothing forcing these molecules to do it. The molecules will just naturally, because of the laws of physics, they're going to move and diffuse according to their own gradient. We can think of diffusion in class, I usually say, like, if you have a bottle of perfume, or like those diffusers for the room to smell nice, or even if you just had like a box of hot pizza, you open up the box, like the high concentration of pizza molecules is right around the pizza box, and you're going to get the smell of pizza immediately. And then the pizza just wafts throughout the room, is going to diffuse throughout the room, right? Those molecules are going to move. And so pretty soon, like, people, in the next room is going to smell pizza, and then 2 rooms down will smell pizza, because the molecules are diffusing. You're not fanning those molecules, you're not pushing those molecules, it just does it naturally. So in terms of the cell, right, this yellow line is going to represent the membrane. Okay, so actually, I'm not going to show you that yet, but if this was the cell membrane, the molecules can just go right, if the molecules are small enough and if the molecules are able to, they can just move right through the membrane down their concentration gradient. Okay, this is actually how your gases of oxygen and carbon dioxide, this is how they move across the membrane. They're small enough and they just whip right through the membrane according to their diffusion gradient. The word facilitated diffusion is the same as diffusion. Molecules will move down their concentration gradient, except they have to have a protein to help them. So this protein is not going to use ATP. Again, the word diffusion is passive transport. But the cells will sometimes put up a protein specifically for a molecule. So water, for example, will move through these, they're called porins, but they're little tiny proteins set up for water. Okay, and they're just open all the time, so water can move in and out without the cell being able to control it. Okay, there are other proteins out here, but when you hear the word facilitated diffusion, it's just that there's a protein set up for molecules to diffuse through. All right, now let's look at osmosis. Osmosis is the movement of water. Specifically, it's the diffusion of water. So number 1, diffusion of water across a semi-permeable membrane. Remember, the cell membrane is our semi-permeable membrane. So we're talking about the cell membrane here. When you talk about diffusion, you have to be familiar with these words: solute, solvent. So solute is the solid particles that are dissolved, like salt or sugar. Solvent is the liquid that does dissolving, and in biology, it's just always water. And then there's the tonicity. When you compare 2 solutions of water, they may have equal or unequal amounts of solute dissolved. You can use the following terms to describe the amount of solute. So let's take a look here at this example before we move on. How do you determine the higher concentration of water? So let's take a look here, right? So we have side A of a beaker, we have a semi-permeable membrane, and we have side B of the beaker. Which side has more solutes? A, right? So the stuff that's dissolved, these red dots, are the solute. So remember, this can be anything. Let's just pretend it's sugar. So this side A has more sugar than side B. Now, we're looking at water diffusing. So if water can pass through this membrane, which way does water want to flow? Water wants to flow from high concentration to low concentration. The more solutes you have, the more stuff you have in the water, the less concentrated the water is. So in terms of just water, side A has less water than B. B has more water. So the higher concentration of water here is going to be side B. Okay, so if B has a higher concentration of water, the high concentration of water here is going to want to flow to side A. Okay, so before we move on to the other side, so we're going to use these terms now. Back to the outline, the word hypertonic, hypotonic, and isotonic. Hypertonic is where you have more solutes dissolved compared to another. So let's use the word hypertonic. Side A is hypertonic to side B. Side A has more solutes in it than side B, right? So side A is the hypertonic side. Hyper meaning a lot or above. So if you have like hyperglycemia, some of you guys may have heard that before, that means you have a lot of sugar, elevated sugar in the blood. So hyper is like elevated. Hypo is below. So you would say side B is hypotonic to side A because side B has less solutes in it. Isotonic is if they had the same amount. We don't have a picture of isotonic here, okay? So let's move on to the other side and see what happens in this. Well, this is just a little practice. So I have 3 different kinds of water for you. I have bottled water, I have tap water, and I have ocean water. So my thought was that this will be our purest example of water. Tap water has fluoride and different ions in it. So maybe it has some solutes in it, but not as many as salt water. Salt water is going to have a lot of salt and different other things dissolved in it. So this will be like the least amount of solutes. This will be in the middle, and this will be the most, okay? So how do we fill these blanks in? Tap water is what to bottled water? Is it hypertonic, hypotonic, or isotonic? The answer is hyper, right? Tap water has more things in it, so it's hypertonic to bottled water. Next example, tap water is blank to seawater. The answer is hypotonic. Seawater is blank to bottled water. The answer is hypertonic. Hyper. 2 bottles of water are blank to each other, and that word is isotonic. Okay, so there's our answers. So that's how you compare the sides. Now, you need to be familiar with those words, and now let's look at the flow of water. Okay, so we have another example. We have solute molecules, right? We have a little barrier here. The solute molecules don't go through, but water can. So osmosis is the diffusion of water. We have a hypotonic solution where we don't have many solutes. We have a hypertonic solution where we have many, many solutes. In terms of water, water will always flow towards the hypertonic side. Always, okay? So water will flow towards the hypertonic side because remember, diffusion means high to low. We have a higher concentration of water here. Water will flow over to the lower concentration of water, right? So the arrow goes in this direction. So what happens is if you let this sit for a while, the water will look like it's getting sucked into the other side, but it's just because of the nature of diffusion. Diffusion, molecules will diffuse to the lower concentration until it reaches equilibrium, okay? So now let's bring it to the cell, okay? Cells are always bathed in some kind of watery environment. There's water inside the cell, there's water outside the cell. The tonicity has to be the same if you have an even flow of water in and out, right? So cells are always bathed in watery fluids. Water flows freely through the membranes. The cell cannot control the water flowing in and out. So most cells must be in an isotonic environment to survive because, okay, not yet. So if you have this isotonic solution, there's no higher concentration of water anywhere. So water just flows in and out, and it keeps the same amount of pressure inside of that cell. However, let's change that up. Let's change the solution. So let's say we put this cell into a hypotonic solution where there's not a lot of solutes. What happens to the water? Remember, water moves to what side? Water will always move to the hypertonic side. So water is going to want to diffuse into the cell, so much so that it's going to want to reach equilibrium. The cell will swell up, right? That's not healthy for a cell. How about the other example here, hypertonic? Now again, water flows towards the hypertonic side. So if you put the cell in a different environment where there's a lot of solutes, the water inside the cell will be sucked out, right? It'll move towards that hypertonic side and the cell will dehydrate. It'll lose water. Also not healthy for the cell. So let's take a look at our scenarios. This is number 3. Osmoregulation is the regulation of water in animal cells, and then we have plant cells. So the first picture is animal cells. Animal cells, remember, don't have cell walls. So let's take a look at these 3 scenarios. This is normal. Water flows in, water flows out. What kind of environment is this normal for animal? Isotonic, right? You have an even flow of water in and out, nothing to the cells happening. If you put the cell in a hypotonic environment, right? Remember that that means that the inside of the cell is the hyper compared to the outside. Water flows in towards the hypertonic side. And if so much water comes in, the cell can break open, it can lyse, basically explode from too much water. Obviously, this is bad. And then the other scenario, right? So this, remember, if you're having trouble thinking about this, it's basically this example here, hypotonic solution. This is the red blood cell. This is the hypotonic solution. Water always flows towards the hypertonic side. Okay. Now this one, you have a shriveled cell. What's happening? You put it in a hypertonic solution. So outside here is very, very high solute. Water flows towards that high solute concentration, towards hypertonic, and you're losing water. The cell will shrivel up. Okay, so that's bad for the animal cell. And this is this example here, the hypertonic solution. Water flows towards the hypertonic side. Okay, now let's look at a plant cell. Plant cells are a little bit different because they have a wall. If a plant cell was in an isotonic solution, the same amount of water in and out, it actually is a little wilty. It's not dead, but it doesn't like an isotonic solution. Plants like to be in a hypotonic, so the water constantly moves into the plant cell. The plant, remember this is called the central vacuole. The plant cell is nice and tight and taut and rigid. The word turgid is used to describe that rigidity. This is where a plant is happy and normal. Then if you have the plant cell in a hypertonic solution, water will get sucked out, okay, move towards a hypertonic environment, and then the cell doesn't shrivel. So the wall of the cell doesn't change. It's the same wall, but the cytoplasm can shrink and shrivel. And that's called plasmolysis. So if you look at your outline, there's a bold word, plasmolysis. That is what you're looking at here. It's where the cytoplasm dehydrates and shrinks. Okay, that's unhealthy. Now, here's a thought. You're making salad. Have you ever made salad, put the salad dressing on your salad, and then walked away? Maybe half an hour later, you come back, and all of the lettuce is now really wilted and, limp. This is what you just did. So the salad dressing is a hypertonic solution because salad dressing has salt and some of it has sugar and there's, different kinds of molecules. And you put the leaf of your lettuce in this hypertonic solution and it's drawing out that water. And so eventually, if you give it enough time, the lettuce becomes very mushy because you sucked out all the water. Another example of this is if you've ever made pie, a lot of pie recipes will ask you to put sugar on top of the strawberries or the fruit that you're making the pie, and you walk away and you come back 15 minutes later and there's a lot of juices that have come out of that fruit, right? The fruit got a little bit softer and now there's water in the bowl. Well, where'd that come from? That's this scenario, okay? You just dehydrated, you pulled out the water from the fruit because you put that sugar on the outside. The sugar is a hypertonic solution, so you can draw out the water. Okay. Diffusion of water is called, hopefully you said osmosis. Okay. Now, I'll pause this and you guys take a look at it. All right, so the answer would be letter C. You add more solute to the left-hand side. So if you just balanced out the solute on both sides of this tube, then the water would be even in both sides, okay? If you place a red blood cell in a hypertonic environment, what would occur? So pause it. All right, so the answer is letter B, water will move out of the cell. All right, so be prepared to answer questions like this on the exam. And now we're going to move on to active transport. So active transport, letter C, is using energy, right? So here's ATP, and we're moving things against their concentration gradient. So remember that diffusion means they're moving down your concentration gradient. Down means you're moving from high to low, like sliding down a slide, high to low. But if you wanted to push something from low concentration to high concentration, like you want to walk up that slide, you have to put energy into that. So if you're pumping, in this example here, right, you have a high concentration of molecules here, you want to add to it, you need to pump that solute molecule towards the higher concentration side, because energetically these molecules want to diffuse down their concentration gradient, right? They don't want to be in high concentration just on one side of the membrane. They want to be equal on both sides. So if the cell wants to keep a high concentration of a certain molecule inside the cell, they need to work at it by pumping in more. So ATP is used. This is a protein pump, and it pumps the molecule to the higher concentration side. Okay, so active transport is the opposite of passive transport. If you look at your picture in your outline, you have that comparison. Okay, so active transport uses ATP to move substances. This should be easy. It should be against, letter B. All right, so this is a recap of all the processes, okay? So we have diffusion, which is just high concentration, moving to a lower concentration. Nothing else is required. These molecules just move from high to low through the membrane. Facilitated diffusion is, again, moving from high to low concentration, but this time we just need a little protein, channel protein, to help the molecules diffuse. Osmosis is the diffusion of water. Okay, so water is going to move from high to low concentration or towards the hypertonic side, right? So we can see that this is definitely the hypertonic side with more solutes than the other side. And then active transport is the opposite. We're moving from a low concentration to a higher concentration. We need to pump that using ATP. So ATP is used. That's what active means. And we're moving against our concentration gradient. All right, so those are the different kinds of membrane transport. Now we're moving on to phagocytosis and endocytosis and exocytosis. So cells will often take in really big things that are too big to actually go right through the membrane or too big for a channel protein. So they'll eat them or spit them out, but in a different way. So let's take a look at, so if this cell here wanted to eat this really big other object, how does it do that? Endocytosis means bringing things into the cell. So letter A under import and export of large substances. Endocytosis is bring substances into the cell. It creates a vacuole by moving the cell's plasma membrane. Vacuole or vesicle, I think vesicle might be a better word. So you're moving substances in by creating a little pocket in the membrane, and then that membrane pinches off to create a little vesicle and it travels into the cell. So this is a nice example of that, right? There's two kinds of endocytosis. There's phagocytosis here and pinocytosis here. Pinocytosis is cell drinking. Just think about Pinot Noir, right? There's a wine, a Pinot. You're drinking Pinot. So when a cell drinks by pinocytosis, it's going to take sips of the water outside. And then whatever is in that water, whatever's floating in that water also gets taken into the cell. So it's kind of like random, whatever, this one didn't even get any molecules. This one got a couple of things. Just whatever gets captured by this little tiny developing vesicles, this pinches off and it brings that substance in the cell. So cell drinking is just taking sips of the water outside. Phagocytosis is cell eating. This one is where the cell will move a larger object, not fluid, into the cell. So this is kind of like this bacteria maybe, and maybe this is a white blood cell, right? That's moving the bacteria inside. This is another example, right? So this is pinocytosis. This is just a little bit more detailed where we see the plasma membrane, the bilayer. We can see the vesicle forming. It's going to fuse eventually, and then we have a vesicle with the substances inside. Exocytosis is the opposite, where a cell might have degraded something inside and you have a lot of waste products to spit out. Or it could be that the cell made something inside, like hormones, for example. You know that your body makes hormones inside of your cells, and then the hormones have to get into your bloodstream, right? So if these were hormones, the cell makes the hormones, packages them up in a vesicle. The vesicle travels to the plasma membrane, they fuse together, and then out go the hormones so it can get into your bloodstream. So that's exocytosis. All right, so this is just a little animation of exocytosis, right? If you were to run this in reverse, it would be endocytosis. And our last topic of this chapter 5 is called cell signaling. So there are a couple ways where cells can kind of know what's going on outside of itself. And this is one way that the cell recognizes there's something on the outside, recognizes something's going on the outside of the cell, but this molecule doesn't have to travel inside the cell, it stays on the outside of the cell. So in this particular example, we have epinephrine, otherwise known as adrenaline, and your adrenaline is made by these adrenal glands that sit on top of your kidneys. Now, adrenaline goes all throughout your body with the bloodstream, and different cells will recognize that there's adrenaline in your blood. But the adrenaline doesn't have to go inside your cells. It can actually stay on the outside in your bloodstream. You have a receptor for adrenaline on your plasma membrane. So these receptors are really important for biology. Receptors are, again, specific to a certain kind of molecule. So this would be an adrenaline receptor. And when it binds adrenaline, basically it signals, it creates a cascade of chemical reactions that eventually leads to some response inside the cell. So this chain of chemical reactions is called transduction, which is why this is called signal transduction. Here's our signal. There's adrenaline on the outside in our blood. There's a chemical reaction going on that's transduced, and then ultimately we have some kind of change in our body, right? So this means that we're breaking down glycogen. Remember, glycogen is that molecule that stores glucose for us. If we break that down, we are releasing energy or glucose into our bloodstream so that we have more energy available to do things like snowboard, I guess. Okay? So that's a signal transduction pathway. All right, so a cell bringing a large solid object into its cell by creating a vesicle is called, your answer should be A, phagocytosis. All right, I think that's it. And that's the end for chapter 5.